Good morning, students. Now coming to experimental proof of semi-conservative method of DNA replication. Experimental proof for semi-conservative method of DNA replication. You know, in previous class, DNA replication it takes place by semi-conservative method. According to Watson Crick, DNA replicates in a semi-conservative method. What is semi-conservative method of DNA? At the end of replication process, one DNA molecule is replicated into two daughter DNA molecules. And daughter DNA molecule is represented by each daughter DNA molecule. It consists of one old strand, means parental strand, and other one is newly created strand. In this way, presence of one old strand and another newly synthesized strands in daughter DNA molecules. This process, this method is called semi-conservative method. Okay. Now coming to the experimental proof of semi-conservative method. During 1958, scientist Michelson and Stahl, they provided a proof. Experimentally proved, these two scientists in 1958 they are experimentally proved the semi-conservative method of DNA. Okay, especially these scientists they proved this semi-conservative method of DNA in Escherichia coli was proved by these two scientists in Escherichia coli. Subsequently, they also proved in human cells and plant cells. Okay. Anyhow, scientists Michelson and Stahl proved semi-conservative method of DNA replication at the first time in 1958 in bacteria Escherichia coli. Subsequently, also proved in the cells of plants and human cells, etc. Okay. Now coming to another point. These scientists Michelson and Stahl, they cultured some E. coli bacterial cells in laboratory conditions and artificially created medium. Okay. Some E. coli cells are cultured on a medium in laboratory, on a medium containing NH4Cl, ammonium chloride. So, these two scientists they cultured some of the bacterial cells in a separate medium containing NH4Cl in which N15 heavy isotope of nitrogen is only the source of nitrogen in this type of medium. So, now this medium contains ammonium chloride in which N15 isotope of heavy isotope of nitrogen is only the source of nitrogen inside the medium. Some of the cells they are grown in this medium. Very important. Scientists Michelson and Stahl they cultured some E. coli cells on a medium containing NH4Cl in which nitrogen is a heavy isotope of nitrogen is the main source of nitrogen compound. And subsequently, okay, now this E. coli is multiplied in this medium by using N heavy nitrogen isotope N15. So, this N15 it is incorporated into DNA. N15 is incorporated into the DNA during the time of bacterial replication. N15 isotope, heavy nitrogen isotope, it is incorporated into DNA, and also other nitrogen compounds. Other nitrogen compounds is incorporated into DNA and other nitrogen compounds. That is this is one experiment. Then you have E. coli are grown in a medium with NH4Cl in which heavy isotope of nitrogen N15 is only the source of nitrogen in this medium. Now this bacterial cell undergoes purification in this medium and it uses N15 isotope of nitrogen for the synthesis of new strands of DNA. So that nitrogen N15 is incorporated into DNA molecules and also other nitrogen compounds present in bacteria. At the same time, these scientists they isolated one of the E. coli cell with a heavy DNA, heavy isotope of DNA, heavy isotope of DNA. That is, the DNA having N15 fibers, N15 and N15 strands of DNA. So hence when this bacteria it is continued biofish in this type of medium, the replication of DNA, it produces new synthesis by using N15 nitrogen, heavy nitrogen. So that the two strands of the DNA molecules present in this type of bacterial cells, they contain heavy isotope of DNA consisting of N15 strand and N15 strand. So N15 DNA is called heavy isotope of DNA. One DNA molecule is isolated from one bacteria with this type of heavy isotope of DNA, heavy DNA is isolated from the medium by using centrifugation. Okay. One bacterial cell with a heavy DNA having N15 strands was isolated from this medium by centrifugation in a cesium chloride density. Okay. One of the N15 DNA is isolated by using centrifugation in cesium chloride density. Okay. Now, this N15 DNA, it is introduced into a normal medium. Now this bacteria with N15 heavy DNA, now it is introduced into a normal medium consisting of NH4Cl. Here nitrogen is N14 nitrogen. Only the source of N14 is available here. So now this bacteria with this DNA is introduced into the normal medium consisting of NH4Cl, in which N14 normal nitrogen is available. That is the next one. Here, okay, this bacteria divides in every 20 minutes once. Bacteria divides by binary fission for every 20 minutes once. The generation time is 20 minutes. In each binary fission, the DNA, heavy DNA, also multiplied into 2 DNA molecules. Okay? In this replication of DNA, new strands are synthesized by using N14 nitrogen. Here, only N14 nitrogen is available in the medium. During the time of DNA replication, for every 20 minutes, new strands are synthesized by using N14 nitrogen. Newly synthesized strands in this medium, they use N14 nitrogen present in available in the normal medium. So at the same time, the scientists they allow this bacteria for bioluminescence in normal medium with NH4Cl in which N14 is there. And also bioluminescence is continued. During the bioluminescence in this N14 medium, scientists also collected the samples for every 20 minutes once, and also they collected samples and also observed what about the strands of newly synthesized strands of DNA molecules. These scientists, they after 20 minutes, okay, what happened after 20 minutes? One DNA is multiplied into two molecules of DNA, two daughter DNA molecules. For example, this is in first 20 minutes, this is DNA. N15 DNA. This is heavy DNA. Now this DNA is incorporated into, it is introduced into the normal medium with the N14. In 20 minutes, this DNA is multiplied into two daughter DNA molecules. First, the two strands, they are separated to each other. This is N15. Two strands are separated to each other. They are heavy DNA strands N15 and N15. Now, by using N14 inside the medium, new strands are synthesized opposite to this old strands. Actually, these two strands of heavy DNA molecules, they are separated and they act as templates in the formation of daughter DNA molecules. This is a template. A new strand is synthesized opposite to the old strand like this, and a new strand is formed opposite to the old strand like this. So after 20 minutes, okay, two DNA molecules are formed. What are the strands present in these two DNA molecules? These two DNA molecules, they contain one old strand, red colored strand. This is template strand and old strand. And blue colored strand is newly synthesized strand by using N14. So one strand is newly synthesized strand, other strand is template strand, means old strand. Both strands are there. This is these DNA molecules are called hybrids. N15 strand. Okay, every DNA is represented by one N15 and other N14. These DNA molecules are called hybrid DNAs. Hybrids. What are hybrids? Hybrids means the DNA molecule, daughter DNA molecules are consist of one old strand and another one is newly synthesized strand. One old and other one is newly synthesized strand. Both are contained in the DNA molecules. That is called hybrid DNA. After 20 minutes, one DNA is multiplied into two DNA molecules and they are hybrids. Hybrids are two DNA molecules are hybrids. So 100%. Hybrids percent is 100%. Okay. And after 40 minutes, what happened after 40 minutes? This two strand, in this DNA, it is multiplied into two DNA molecules. It is also multiplied into two DNA molecules. First, two strands are separated. This is old strand N15, and this is newly synthesized strand N14. Two strands get separated to each other. Here also, two strands they separated to each other. This is N15 and this is N14. In another 20 minutes, that is after 40 minutes, these two DNA molecules they are multiplied into four DNA molecules. First, these two strands are separated to each other. These two strands are separated to each other. Now, these strands they produce opposite new strands. By using N14, it is developed into one DNA. By using N14, develops into one DNA. By using N14 in this way, N14 strands are formed opposite to these old strands, leading to the formation of total 4 DNA molecules after 40 minutes. So here, how many DNA molecules they contain? One N15 and 114. How many DNA molecules contain both N14? The DNA molecules, for example, hybrids. Here, hybrids 2. This is hybrid N14 N15. This is also hybrid N15 and N14 and N15. There are total 4 DNA molecules. 2 DNA molecules are hybrids. And how many DNA molecules are light DNA molecules? Light means both strands are N14. Both strands are N14. This is light. This is light. This is hybrid. This is light. This is hybrid. This is light. And light DNA also 2. What's the percentage of hybrids after 20 minutes after first multiplication, first generation? 100% hybrids. But what about the hybrids percentage after second phase? Hybrids are reduced into 50%. Only two hybrids are there and 2 light. And what about after 50 minutes, after 60 minutes? After 60 minutes, what happened? 4 DNA molecules are multiplied into total 8 DNA molecules. And how many are hybrids and how many are light? Only 2 are hybrids in any generation. Only hybrids number is 2. And all the remaining DNA molecules, they are the light DNA. After third generation, 8 DNA molecules are formed. Total 8 DNAs are formed. Here 2 hybrids and 6 light. After 1 hour 20 minutes, that is after the fourth generation, 16 DNA molecules are formed. 2 are hybrids, 14 are light. After fifth generation, 32 DNA molecules are formed. 2 hybrid, 30 are light. Hybrids number is constant, that is 2. And remaining DNA molecules, they are formed by using N14 medium, N14 nitrogen, so that all of them, they are light DNA molecules. Like that. Now coming to a simple table that explains the generations and light and hybrids percentages in generation to generation. Very, very important in objective purpose. Now coming to one. This is a small table that explains the proportions. Okay. First column for number of generations. Number of generations. Okay. And second column for time in minutes. Time in minutes. Okay. Third column for total DNA. Total DNA. And fourth column for hybrid DNA. Hybrids number. Okay. And next column for number of light DNA. And another column, this is for percentage of hybrids. Percentage of hybrid DNA. So that is the table. Now coming to one by one. Here after first generation. Okay. After first generation, Time, that is 20 minutes. First generation 20 minutes. Okay. How many DNA molecules are formed after first generation? One DNA is multiplied into two DNA molecules. Two DNA molecules. Okay. And how many hybrids are there? Two DNA molecules are hybrids. No like DNA. Like DNA, no. Percentage of hybrid DNA 100%. This is the first generation. In first generation, within 20 minutes, one DNA is multiplied into two daughter DNA molecules, and two DNA molecules are hybrid DNA molecules having one N14 and the other one is N15. Here, in first generation, there is no the like DNA's number, and percentage of hybrids are 100%. Now coming to the second generation. That is in 40 minutes. Okay. Total 4 DNA molecules are formed. Two DNA molecules are hybrids. Remaining two DNA molecules are like DNA molecules, and 50% hybrids. This is after 40 minutes. That is after second generation. Now coming to third generation, that is after 60 minutes, 8 DNA molecules are formed, 2 DNA molecules are hybrids, and 6 DNA molecules are like DNAs. Here also hybrids number reduced to half, that is 25%. And in fourth generation, that is 80 minutes, 16 DNA molecules are formed, 2 are hybrids, and 14 are light, and 12.5%. And now coming to another generation, this is fifth generation, that is after 100 minutes, and 32 total DNA molecules are formed, and 2 DNA molecules are hybrids, 30 DNA molecules are light, and 6.25%. Okay, very important. This table is applicable in, that is when very important, when a heavy DNA having N15 into a normal medium having N14 nitrogen. This is the table. In which situation? When a heavy DNA having N15 is transferred into a normal medium, that is with N14. This is the proportions of various DNA molecules. Very important. Okay. Now coming to another point, very important. Another scientist. It was discovered by scientists Michelson and Stahl in equally. Semi-conservative method, it was experimentally proved by Michelson and Stahl in equally first. Later it was also explained in human cells and also plant cells. That is in bacteria. It was explained in bacteria. That is a semi-conservative method of DNA replication. At the same time, another scientist Taylor also explained semi-conservative method of chromosomes. Explained in chromosomes, especially in Vicia faba plant. Vicia faba. Okay, faba bean. This is also called faba bean. By using radioactive thymidine, very important. Radioactive thymidine. Actually, semi-conservative method in of DNA replication in E. coli was discovered by these two scientists. At the same time, the semi-conservative method of chromosomes was discovered by scientist Taylor in Vicia faba bean. Here, he utilized the radioactive thymidine. Radioactive thymidine was utilized by scientist Taylor.